and I welcome you all for your gracious presence, physically as well as virtually, for attending this seminar. I'm pleased to introduce Ms. Krishnashilpa, who is one of the Ph.D. research scholar and who has worked in biotic pre-breeding lab for her Ph.D. thesis. She has done her BSc and MSc in microbiology from Sri Krishna Dev Raya University, Anantapur, and for PhD she is registered with JNTU, Hyderabad for biotechnology. She is here to share with us the research findings on the topic development of synthetic tetraploids, new sources of tetraploid groundnut. So this is over to Shilpa now.
Thank you, ma'am. Here, my topic of research is development of synthetic groundnut or a new source of tetraploid groundnut. Before going directly to the development of synthetics, I just want to talk few words about the cultivated groundnut and the reasons which made it path for the development of new source of groundnut. Coming to the introduction, groundnut is an tetraploid widely grown in tropical and subtropical regions of the world. More than 100 countries cultivate groundnut and has an average production of 35.5 million tons annually. China leads in the production of groundnut, followed by India, United States, and Nigeria. As I said, groundnut is an important crop, oilseed crop in India. If we observe the map, the light brown color indicates the minor production of groundnut and dark brown indicates the major production of groundnut. If we observe the Gujarat, groundnut is one of the major crop. Now coming to the nutritional facts of groundnut. Groundnut is rich in nutrients and more than 30 essential nutrients are present, which include niacin, folate, magnesium, vitamin E, etc. A plumpy nut ready to use is a therapeutic food prepared from the groundnut. It is used by UNESCO to treat acute malnutrition and acute malnutrition kids in Africa. In groundnut, it consists of more than 25% protein than any other true nut. Coming to the scientific classification, it belongs to kingdom Plantae, division Tracheophyta, class Magnoliophyta, family Fabaceae, genus Arachis, and species Hypogaea. There are 80 species in genus Arachis which have been divided into 9 different sections based on the crossability and few morphological traits. Coming to the genetic variability in genus Arachis, Hypogaea and Monticola are in primary gene pool. The secondary gene pool includes diploid Arachis species in section Arachis, and in tertiary gene pool includes the species which are in section Procumbens, and remaining 7 will come in quaternary gene pool. There are several Arachis species cultivated, but Arachis hypogaea is the only one domesticated, widely distributed for commercial production of seed and oil. But this Arachis hypogaea, it is having narrow genetic base, probably because of the bottlenecks associated with its origin. Coming to the origin of cultivated groundnut, when 26 cross-compatible species are available in groundnut, only 2 species, Arachis duranensis and Arachis hypogaea, are the putative donors for the formation of Arachis hypogaea. As I said, the cultivated groundnut, because of its narrow genetic base, it is susceptible to many disease and pests. But on the other hand, the wild Arachis species, they are resistant to many diseases and pests. Coming to the genomes in Arachis, initially they are A, B and D genome, but recently Robledo scientists have identified another 2 types of genomes, K genome and F genome. Coming to the chromosome structure, A duranensis, in A duranensis we find larger heterochromatin band in the centromere, and in Ipiensis, if you see the chromosome structure in Ipiensis, heterochromatin band is not there. In Batizochi, we again find a heterochromatin band formation. Initially, they thought that duranensis and Batizochi are the putative donors for Arachis hypogaea, but based on the restriction fragment length polymorphism and fish mapping rDNA loci, they confirmed that Arachis duranensis and Arachis ipiensis are the putative donors for Arachis hypogaea. So the species which are having the chromosome structure similar to A duranensis are categorized as A genome, and showing the species which are showing the chromosome structure similar to A ipiensis are categorized into B, and Batizochi, they are categorized into K. I'm not going for F and D because I'm not having that chromosome study. So here what I said, the wild Arachis species are having resistance when compared to that of cultivars. The scientists are anxious to to produce new types of synthetics to overcome the drawbacks of the narrow genetic base. For that we need more diploid hybrids, more tetraploids for development of tetraploids. But one of the major bottleneck is interspecific gene transfer because of the difference in the ploidy between the diploid and cultivated groundnut. So there are many methods and many pathways have been developed by scientists to overcome these, and they are successful in transferring the gene of interest into the cultivar. Based on this background, ICRISAT has taken initiative in development of new source of tetraploid. Dr. Nalini Mallikarjuna Madam has developed nearly 17 different types of synthetics belonging to different genome species and belonging to the different species and belonging to the different genome wise. Now coming to the development of synthetics. In development of synthetics, most recently the crosses were made between synthetics for developing the double synthetic. Here, in the double synthetic one, we have selected ICRISAT-GR1212 a synthetic and ICRISAT-GR9A. Coming to the background of ICRISAT-GR1212, a cross was made between Juranensis, which is a genome, and Ipiensis, which is a B genome. At the same time, ICRISAT-GR9A further, a cross was made between Cardinaceae, which is A, and Batizochi, which is a K genome. So in synthetic ICRISAT-GR1212, AB genomes are there, and in synthetic ICRISAT-GR9A, AK genomes are there. A total of 189 pollinations were done, out of which 40 pecks are formed and 16 pod formation was seen. So for confirmation whether the double synthetic or hybrid or not, if we observe this, the peak 90 has come from 9A and 400 has come from 121, ICRISAT-GR1212. After confirming that, we extended our work with cytological analysis. Steps involved in cytological analysis are collection of birds, bud fixing, dissection of birds, pollen fertility and analysis. Collection of floral birds. Birds are collected from glasshouse at a particular time. They say that at a particular time 7:30 to 8:30, but we collected randomly, but we are important upon the size of the bird. Later they are fixed in corner solution 2 for 2 days and followed by corner solution 1 for 1 day, and they are dissected for anthers, and the anthers are squashed in acetocarmine for further analysis, and pollen fertility is also analyzed using acetocarmine. Now cytological analysis of double synthetic 1. If you observe the picture, in the cytological analysis, the first two are metaphase, anaphase, tetrads and pollen. If you observe the first photograph, here all the 20 bivalents are seen, whereas in this few univalents also, you can easily see the univalents, presence of univalents. In the second, it is anaphase. In anaphase, equal distribution is seen, whereas here we can find some laggard formation. Coming to the tetrad analysis, in tetrad analysis there is no formation of pentads or diodes, but the formation of triads and tetrads are there. So coming to the triad percentage, 1.3% of cells are showing triad formation without any micronuclei, but tetrads are having micronuclei. 2.4% of tetrads are having single micronuclei, and 1.2% are having double micronuclei, and remaining are the normal tetrads. There are abnormalities in cytological analysis, but the percentage of abnormality is very few that didn't affect the pollen fertility. If you observe the pollen fertility slide, in this the dark pink and the round color, they are fertile, and small which are light pink, they are sterile. If you see the slide, the pollen fertility is high, it is 91%, and because of the good pollen fertility, good pod formation is seen. Now coming to the development of double synthetic 2. Here also we have selected ICRISAT-GR1212 as a female parent, but we have changed the male parent and it is ICRISAT-GR5B. Coming to the background of ICRISAT-GR5B, it is made up of Batizochi, Magna into Batizochi. Again, Magna is a B genome and Batizochi is a K genome. So here ICRISAT-GR1212, it consists of AB genome, and 5B it consists of BK genome, and the double synthetic developed consists of all A, B and K genome. Coming to the peak pictures which are taken from the GeneMapper software, here if you observe 391 has come from 5B and 400 is come from 1212, and these both peaks are present in ICRISAT-GR1212 into ICRISAT-GR9A, which is a double synthetic 2. Coming to the cytological analysis in double synthetic 2, here also few abnormalities are observed. In the first slide, first two pictures, here we can easily see the univalents. and 20 bivalent formation is observed. Here univalents are formed. Here one trivalent is seen. Here the equal distribution, and if you see this, there are the laggard formation. There are more than 5 to 6 laggard formation. Now coming to the tetrad analysis. In tetrad analysis, micronuclei are present, triads are present, but the percentage is very less, the same, that didn't affect the pollen fertility, and it has resulted in the good pollen fertility, good pod formation. Coming to the double synthetic 3, here we have changed the female plant. It is 278-18. Coming to the background, it is developed from Duranensis into Batizochi, but whatever the Duranensis, Batizochi used in the 5B is a different accession number, and this is different. So here also the Batizochi and Duranensis, it belongs to A, and Batizochi belongs to the K, and pollinations were done. Nearly 189 pollinations were done. Out of 26 pecks are formed and 15 pods are formed. Coming to this is again a peak picture. Here, if you see, the 170 has come from 278-18 and 185 has come from 5B, and both peaks are present in 278-18 into 5B. So if you see the cytological analysis, here also we find few abnormalities, but the percentage of abnormality is not more. So that resulted in a good pollen fertility. And very interesting thing of this double synthetic is double seeded pod, which is not seen in any Arachis or wild species till now. This is a special character of this double synthetic 3, the double seeded nature. Now we extended our work from cytological analysis to the molecular analysis using SSR marker analysis. Steps involved in SSR marker analysis, it includes isolation of DNA by CTAB method, checking the quality of DNA on agarose gel, running PCR with the selected markers, amplified products were subjected to capillary electrophoresis, scoring was done on using gene scan software, and the score was subjected to Darwin and power marker. So this is the first, it is the DNA what we isolated from CTAB, and we need only 5 to 10 nanograms of DNA concentration to run PCR, so they are diluted with distilled water. Coming to the primers, we selected 110 primers from 20 linkage groups based on the series of a map, and if you see that some or amplified, some primers amplification is seen and in some amplification is absent. So coming to the individual scoring of results of SSR markers for individual genome wise, if you see here the polymorphic primers, in a genome a total out of 71, I said as 120 primers have used, out of 71 are polymorphic, but when we come for an individual scoring, in a genome there are out of 71, 51 are polymorphic, and they are detected altogether they detected 147 alleles with an average of 2.5 per marker. Coming to the peak value, it is 0.51, and genetic diversity, it is 0.61. Now coming to the B genome. In B genome, out of 71, 52 markers are polymorphic and alleles detected are 170, and with an average of 3.2 per marker. Coming to the peak value, it is 0.3 and 0.53, and genetic diversity is 0.63. Now coming to the synthetics, out of 71 in synthetic, 57 are polymorphic, and they score 250 alleles with an average of 4 per marker. Coming to the peak value, it is 0.54, and genetic diversity, it is 0.62, whereas in double synthetic, it is more than 67 are polymorphic, and we observe 462 alleles with an average of 6.5 per marker, and genetic diversity, it is 0.71, and genetic diversity is 0.77, and genetic peak value is 0.171. If you see the cultivars, in cultivars total 70 polymorphic primers are detected and 350 alleles are detected with an average of 5.6 per marker. Peak value, it is 0.60, and genetic diversity is 67. If we observe here, the alleles are more in double synthetics than any other, and even the genetic diversity is high in double synthetics, and peak value is also high. Peak value is a polymorphic information content. So here, by this SSR marker analysis, we are concluding that they are diverse. Double synthetics developed are more diverse. The scores, we subjected the score from gene mapper software to Darwin software. Here we get a dendrogram. From the dendrogram, if you see that ICRISAT 1212 into ICRISAT 9A, it is grouped with TXAG6. If you see the background of TXAG6, it consists of are Cardinaceae into dicot, which is again crossed with Batizochi. So there are total of Cardinaceae and the Batizochi are also present in 1212 into 9A. So that may be the reason that have been grouped them in a, that has made them to group together. On the other side, if you see the TMA, there is no parental relationship, but they grouped together. From the dendrogram, it is clear that there is a cross compatibility between AB genome, AK genome. So here the wild species which are green, which are in green color, the grouping is formed based on the smaller dissimilarity. They are having, which are having less dissimilarity, they are grouped. Now coming to the next step, protein inhibition assay in synthetics, double synthetics and two cultivars. The steps involve crude protein extract from the seeds and protein estimation using L'Oreal's method and using BAPNA as a substrate. Their trypsin inhibition assay was done, and using gluferra as a substrate, chymotrypsin inhibition assay was done. Coming to the results of proteinase inhibition, if we see here, the TMV2, which is a cultivar, is having high trypsin inhibition value. We can say that proteinase inhibitors. But in synthetics, they are, if you compare between TM2 and JL24, the synthetics are having more than JL24 but less than TMV2. But the double synthetics, they are almost equal to the synthetics, cultivars. Coming to the chymotrypsin, it is not at all detected in TMV2, but JL24 it is having some, JL24 is having that chymotrypsin proteinase inhibitors. If we see the synthetics and double synthetics, all are having the proteinase inhibitors. Now the next topic is development of epicutial population, which was developed by our Madam Dr. Nalini Mallikarjuna. So here, coming to the background, here we used 278-18, we crossed with the cultivar ICGV 87846. Coming to the background of 278-18, it was, a cross was done between Juranensis into Batizochi, and that resulted in a diploid hybrid which has treated with the colchicine and it has converted into tetraploid. This tetraploid is crossed with ICGV 87846. The F1s which they are involved in the first back rows, and they did two back rows, and finally the final obtained they subjected for advanced screening. In advanced screening is done for by infector row method. In this technique, the genotypes of epicutial population were shown in two replicated plots of same size, that is, each plot is having 32 lines. In each test plot between 4 rows of the test variety, the cultivated groundnut, which is susceptible to LLS and rust. If we see here, this is the susceptible line, and these are the 4 lines of the test epicutial population. In between 4 lines, ACGV 87846 was planted. This is because they are susceptible, and the conidiospores which are released, they can easily spread, originospores released by them, they can easily spread to the adjacent plant. If the plant is susceptible, they show the symptoms of disease. Here are the variations which we observed in population 3. If we see the photograph, here it is bunch with orange flower, spreading type with yellow flower, semi-spreading with orange and semi-spreading with yellow flowers. These are the variations what we observed in the field. In the same way, the variations are also observed in the pod. If you observe the photographs, there are variations like slight beak, moderate constriction and moderate reticulation in the first photograph. If you see here, very prominent beak, very deep constriction and prominent reticulation. Here, deep constriction. Here I am saying very prominent deep constriction, and here deep constriction. There are the variations between the pods which we obtained from the population 3. And if you observe here, the beak is absent, but deep constriction is there, reticulation is absent. The same way here, the beak is absent, slight reticulation and slight constriction. All these are the variations which we observed in population 3. Coming to the disease symptom, this is the susceptible plant which is showing the disease symptoms of LLS and rust. But on the other hand, this is the epicutial population which is not showing any symptoms of plant. See, if you see the score, majority of lines in LLS, this is the score taken on the 90th day. Majority of lines they are having between 2.5 to 3, indicating that they are resistant. On the other hand, the rust score, it is 2, indicating they are resistant in nature. See, if you see that this is, we have taken the score based on the 9.9 modified 9-point scale. In this, if you see the score 3, the symptoms are appear at the lower side of the plant, and small few amount of spots are appeared in the middle, and a slight defoliation may occur, but the 3 indicates they are resistant. Coming to the results of proteinase inhibitors in population 3, we also did proteinase inhibition assay in population 3. If we observe the first 2 are the parents, the protein estimation is high when compared to that of first 2 parents. Coming to the trypsin inhibitor assay, the proteinases, here also we find first 2 are the parents and remaining are the ubiquitous reels. Here we find the maximum of 8, which is not seen in any other, indicating thus they are having high resistance to Insectodoptera. Chymotrypsin assay. In chymotrypsin assay, even though there is not much variation, but proteinase inhibitors, it indicates that proteinase inhibitors are present. In the same pattern, we have collected gut from the insect and we subjected for the gut inhibition assay. Here also the gut inhibition assay is showing more than what in parents, indicating that they are insect resistant. Coming to the fatty acid composition in synthetic tetraploids. Fatty acid composition is important for any oil yielding crop because it decides whether the oil is healthy for human consumption or not. So, groundnut oil is considered as one of the nutritious oil because it is having high amount of TUSF when compared to that of TSF. Coming to the oleic acid. Presence of oleic acid hinders the adrenoleucose dystrophy, which is a fatal disease of brain and adrenal glands. Here we can see that 37% to 43 in cultivated, and synthetic it is 33 to 43. There is no much variation between the cultivated and synthetic. Coming to the linoleic acid deficiency causes scaling of the skin and hair loss, and here also if you see there is not much variation between the cultivated and synthetic tetraploids, and remaining also if you see there is not much variation between cultivated and synthetic tetraploid. Coming to the iodine value indicates the unsaturated nature of the oil. More iodine value indicates more unsaturation with more double bonds, and less iodine value indicates less unsaturation and with less double bonds. But here if you observe that synthetic tetraploid, it is having 88 to 105, whereas the highest is seen in 130 in coconut. But from this result, we can consider that the iodine unsaturation nature is more in groundnut. Unsaturated nature of oil is good for health. Coming to the conclusion, meiotic analysis of double synthetic showed minor variation in chromosome configuration in metaphase, anaphase, tetrads, but it resulted in high pollen fertility. From SSR analysis, it is clear that genetic diversity and peak values are more in double synthetics when compared to the synthetic cultivars and wild species. Coming to the protein inhibition assay, proteinase inhibitors are present in double synthetics and also in developed abiotic population. Successful introgression of LLS and rust resistance gene from wild to cultivated to abiotic population. Here because the score is 3 in rust, LLS and 2 in rust, we can say that this population is LLS and rust resistant. Finally, the fatty acid composition. From the result of the fatty acid composition, it is clear that groundnut is healthier than any high degree of unsaturation when compared to that of saturation. Coming to the acknowledgement, with great pleasure express my esteemed gratitude and deep sense of indebtedness to my respected supervisor, Dr. Nalini Mallikarjuna Madam, Principal Scientist, Legumes Cell Biology, ICRISAT, whose help, stimulating suggestion and continual encouragement has helped me to all the time in my research and also for writing purpose. My special thanks to Rosanna Mullah Madam, Coordinator, Learning System Unit, permitting me to avail the research and use the library facilities. I would like to express my sincere thanks to Dr. Manish Pandey sir and Dr. Pawan Kera sir for their suggestions and guidelines regarding the molecular work in AGL. And I like to thank my friend Sri Swati for helping me. She is the girl who helped me in learning all the softwares in AGL department. I would like to thank my friends Sandhya and Samira. Last but not least, I would like to thank our supervisor Shivali Sharma for all she did for us. Thank you.
Now the seminar is open for
Okay, Sandhya, just can you tell that of the 16 synthetics or 15, 17 synthetics, so which one you will suggest that is good for disease resistance, and the one which one is good for, like, say, other traits, say some pod traits or some other traits?
If we see that coming to the disease resistance and other traits, so in disease resistance, if we see the synthetics, what we coming to the other traits, see here we have developed epicutial population which is having the synthetic 278-18. From the result we can say that that plant is resistant to LLS and rust. Coming to the other traits, coming to the trypsin inhibition assay, I'm telling the what we did from the practical what we did. Coming to the trypsin assay, coming to the trypsin assay 1212 and 10B is having 17 to 18.9 units per mg protein. That is trypsin inhibition activity, TI activity. Cultivated groundnut, when we and the trypsin, coming to the CI unit, it was not at all detected in some cultivars, but it detected in few. But in double synthetic, if you see, in double synthetic, it is 17 in 1212 and 9A. I said ICRISAT 1212 in 9A, the 17 units are there, and it is the highest unit when compared to the other cultivars. So we can say that the double synthetic is having high units of trypsin and proteinase inhibitors belonging to the trypsin. And coming to the other traits, like I did in synthetics, I just did the trypsin activity because developed from Nalini Mallikarjun ma'am. I did only trypsin activity. From trypsin activity, I can say that 1212 and 9 are having maximum.
You also told about double podded or
Double podded nature is in double synthetic 3, which is developed from 278-18 into ICRISAT 5B.
And that is not there in either of the parents.
That is not in either of the parents.
Okay, so these lines are stabilized lines.
This line, now it is in F3 generation. It should be some, if some more generation follows, it stabilizes. In the F3, F2 generations, we find the double-seeded nature. In F3 also, we find double-seeded nature of the pods.
So from the crossing with like different wild species, do you think that using other except A and B genomes, I'm telling, except A and B, you have used other species as well, right?
Yeah, we used A, K, A, B,
yeah.
B,
So
K.
how is their performance?
That from the dendrogram, I've said that the cross compatibility between A, K, A, B and B, K is good. That is the grouping system in dendrogram, they are grouped together, and if you see from the pollen fertility, we can say that they are compatible, cross-compatible. AK genome, AB genome and BK genomes are cross-compatible.
So do you think that if synthetics may be already developed, or in future if it is developed from these combinations, they can be directly used with the cultivated genotypes, right?
Once they are stabilized, we can use this cultivar.
Okay.
Shilpa, in the epicutial population, when you screen for the rust resistance, almost all the genotypes have the score of 2. Unlike observed in a single segregating population where the score varies from individual to individuals. What could be the reason behind the same?
Here we have taken the mean value of all. We are not taken individual plant. We have taken the entire mean value of the plant.
Segregation means it will come like higher, also it will go 6, 7. So how much highest will you receive here? Like
In highest we received till 5.
maybe not severe disease like symptoms you observe in that particular.
No.
Maybe it was like what was the score of this in the parent?
In parent, if we check in cultivar, it is 7 to 8. Nearly 90% of destruction is there. whereas in 278-18 it is 4.
So you crossing 4 with 9 and getting almost all as 2, not surprising. All are not getting it with the
Shilpa, I have a question for you. You said double synthetics. How many double synthetics you have developed?
3.
So they are in F3 generation. Would you please tell me how many seeds are available for each of these 3 crosses in F3?
F3, ma'am, or whatever we did for trypsin inhibition assay, the remaining we have given to the department.
No, how many seeds?
There are for a double synthetic one approximately 10 pods. And
Only 10 pods. Wait. 10 pods for double
Double
synthetic
synthetic
one.
one.
Per second.
For second, it is 10 because our Nalini ma'am has given me a 20 pods for my practical work.
Because when I received the packets, there is only 1 or 2 pods. That too when you talk about F3 generation. So this is again for Manish Pandey also, as you told that once they get stabilized, we can use them. But we have only 10 pods, and in one cross we have only 5 pods, and one packet is still empty because they have used in trypsin inhibitor
When
and
Nalini
other
ma'am
things.
is there, I got only the pods for my work. At that time, I'm doing trypsin inhibition
But
assay.
where are those F3 generations? As you are saying that they are in F3 population.
That-
Where are those F3 generations?
F3 packets, whatever I received, I've given to you.
So, but those are empty. I can show it now.
No,
There are only 2 pods and 1 packet.
no, 1 to 1, ICRISAT 1 to 1 to into 5B, they are very less pods, and that I completely used it. And
That's
there are
what
1 to
we want to
2
listen
pods.
from you because we don't have the material.
Remember,
And you are
I just
saying
taken the pod.
Yeah.
No,
Think.
I
Be
just
very
taken
clear.
for my work.
Yes. No, but that's what I'm saying. How many pods you have returned to me?
Okay.
Tell me now. Well, how many pods you have returned to me for double synthetic 1, 2 and 3? Because this is something
3
very
is
precious
almost
material.
10 to 15.
Okay, 10 to 15 pods. Okay, and yes, double synthetic 1,
Double synthetic 1 10 now.
10 and 2?
2 we are not having.
We are not having. Okay, and these 20 in together, 20 pods, they are in F3 generation. You have developed the double synthetics, and now you are also aware of wild diploid species which are crossable. And then we have synthetics which are again crossable. So we have 3 kind of resources: double synthetic, synthetic, and then simple synthetics and double synthetic. So what do you think, or what is from your perspective, that which should be the best resource to utilize the maximum variability or the diversity for groundnut improvement program?
See, because there is a ploidy difference between the cultivars and diploid species, direct intraspecific gene transfer is not possible. For that, we need to develop synthetics. So if you go to the background of synthetics, it takes more time. Like we have to cross between diploid, we have to treat with the colchicine, and we have to get the synthetics. Sometimes even after colchicine treatment, they may revert back and they form diploid. But in double synthetic, when compared to the synthetics, it is a faster route because we are directly crossing between synthetics. We are getting tetraploids, direct tetraploids. In synthetics, we have to wait. That is a time-taking process. When synthetics are available, when we cross between 2 synthetics, the time-taking process is less and we can get a good quality. As we said in double synthetic 3, the double-seeded pod nature. The double-seeded pod in nature was not at all observed in any Arachis species and in any synthetic till now.
In synthetics, we have double podded.
In synthetic, not in double synthetic.
No, but in synthetics we have, because to develop this double synthetics, we have first to generate the synthetics. So the route remains the same, whether it's synthetic or the double synthetic. So double synthetics is nothing but the cross between 2 synthetics, F1. So do you think it is easier to use synthetic directly for groundnut improvement, or it is much easier to use double synthetic for groundnut improvement? Because we want to accelerate
I can say that
enhance the efficiency of
in synthetics we have only 2 genomes, A or B or A anyway. But in double synthetics there are 4 Arachis species. From 4 Arachis species we can get more gene of interest when compared to that of synthetics.
What will you suggest us? How many crosses do we need to make when we have double synthetic, where we have 4 genomes together, because every species has a genetic load? So what do you think? What do you suggest? How many F1s shall I make when I want to use double synthetic as a male parent?
Double synthetic, if we use a double synthetic, when we cross, we get in the first F1 generation we got 10 to 15 pods.
No, my question is, when we have the stabilized source. One is for stabilization of double synthetic, it will take time.
Yeah.
Once we have, what do you think? Because in your case, you have developed only for the development purpose, you have done 189 pollination. So when we want to use double synthetic for groundnut improvement program, how many F1C do we need to have to exploit the full diversity of 4 genome species which is present in double synthetic?
When we do, we can say.
No,
100 is required.
no, because when we talk about 4 species and to represent each and every species, I don't know. So what is your take on that?
Like all the 4, I'm saying that all the 4 species are there, but in F1 we can, depending upon the cross compatibility between the species, because there are 4, depending upon the cross compatibility we can get. Suppose if there is no limit for any number in that.
So any synthetic like only it has only A and B genome, double synthetic. Any double synthetic is like developed from 2 synthetic, they have only A and B, not others.
No, initially when we scientists are classified into A, B, D genome.
Right.
At that time, Magna and Batizokai they come under B genome. That time they are called as autotetraploids.
Later they proved
But
to be
they
K.
proved that K and it has become AK, BK.
So whether it is developed in US, you are telling about that.
Fish mapping based on the fish mapping RDNA loci by W. scientists, 2010.
They utilized those ones and
They did the fish mapper
they
analysis.
reported something.
They did the fish map RDNA loci and they identified that if you see the chromosome structure, the heterochromatin is, see, if we see here, they reported that here if you see that Juranensis and Batizokai, they are having heterochromatin, but they are not equal based on the, if you see the dark parts, they are 18S and 23S RNA, and black and white lines indicate 5S RNA. Based on this, they have identified that the genomes which are having similar structure to Batizokai, they are categorized into K. On the other side, the F genomes, they are having heterochromatin, but in B they are having larger heterochromatin band, but in F we find only tiny bands.
Okay, so you have developed how many double synthetics?
3 double synthetic.
So of the 3, which one you will suggest that will be more useful to develop?
Yeah.
Which one?
Which
You
one?
like
Double synthetic 3. Because why? Because
what genomes
it
it has?
has from the diploid species it is having 2 synthetics, 278-18 into
Yeah, that is the
ICRISAT.
good one. Yes, from
But-
Harvard also they reported.
Because double-seeded nature is seen only in that double synthetic. Even this is resistant
Better in 278.
278-18. Double-seeded nature, this is because it is not observed in any Arachis species, that will be the best quality of double synthetic.
Okay, so you give that name, something you give name to that double synthetic.
Double-
Like ISATGR, we heard for synthetics like ISATGR.
1212 into ICRISAT.
For double synthetics, how you named them? Because
We-
there should be some nomenclature.
We named as double synthetic 1, double synthetic 2 and 3.
So just maybe if you are putting in your thesis, if it is not finalized, give some proper name.
Okay.
If you are going for publication also, give proper name. We just discuss like what kind of nomenclature, not even this crop you see in other crops also like synthetic.
Okay.
So give proper name
Okay.
to them so that they will be recognized with the name. So you don't know to do.
Okay.
Yeah. And in your thesis and everything, whenever you are publishing or publish something, I hope you will find that these are the best ones. Something should come from your studies.
Okay.
Have you studied any differences between last resistance and more synthetic
Everybody?
studied some differences in between your developed synthetic and cultivated
Actually, synthetics, they are developed by our manufacturing animal manufacturers. My company is developing of synthetics. We develop and the people who follow that, they may go further study. Because they are yet to stabilize.
Resistant like that synthetic, getting some synthetic in, then it should be in your yield at par or some higher yield in that.
Now we are actually using the synthetics for introgression of resistance. So another way we have developed the epithelial population. by taking one synthetic and crossing with the hatch.
Do any time the dormancy was tested?
It was a problem in any of these problems. Say dormancy was observed in any of these.
No, sir. Because in one topic we didn't find any dormancy.
When we multiply the seeds, there is definitely a problem of seed germination, and some were identified as rejected because they did not germinate. You can check these problems with Shivali. Any dormancy work on these synthetics, because we have some problems in seed multiplication, especially the crosses, which easily comes from wild nature.
The problem is I think around 5 or 6, and last time they were multiplied, it was in 2008, and those were just those were not actually stored properly. So we also face very high problem of poor seed germination. I think Dr. Upadhyay were given 100 pods and none of them germinated. So we also had a similar problem. So it is not because of the dormancy. This is because of the poor storage of those synthetics. Of the double synthetic, we don't have any seed.
Right?
Double synthetic you have
So
it.
what I did, we can do the parentage, if you parentage are known, so maybe you can make an effort to make
double
again
synthetic
double
3
synthetic.
you have it for.
You have pots?
Yeah.
Which time is pots are there?
Two.
Because what I'm thinking, if it is important, so because once you know that these parents are good for me, they are coming good, they are bringing good trade, then you can make it again, because every time it will may be different. Because those are segregating, no? So each cross may be different. So that will be good.
Any other question?
Anything?
So let's give her a big hand. Thank you very much.